Polymerase chain reaction, or PCR, is a common laboratory technique used to make many copies of a particular region of DNA. This DNA region can be anything the experimenter is interested in. For example, it might be a gene whose function a researcher wants to understand, or a genetic marker used by forensic scientists to match the crime scene DNA with suspects. Typically, the goal of PCR is to make enough of the target DNA region that it can be analyzed or used in some other way. For instance, DNA amplified by PCR may be sent for sequencing, visualized by gel electrophoresis, or cloned into a plasmid for further experiments. PCR is used in many areas of biology and medicine, including molecular biology research, medical diagnostics, and even some branches of ecology.
Like DNA replication in an organism, PCR requires a DNA polymerase enzyme that makes new strands of DNA using existing strands as templates. The DNA polymerase typically used in PCR is called Taq polymerase, after the heat-tolerant bacterium from which it was isolated, Thermus aquaticus. T. aquaticus lives in hot springs and hydrothermal vents. Its DNA polymerase is very heat stable and is most active around 70 degrees Celsius. This heat stability makes Taq polymerase ideal for PCR. As we'll see, high temperature is used repeatedly in PCR to denature the template DNA or separate its strands.
Like other DNA polymerases, Taq polymerase can only make DNA if it's given a primer, a short sequence of nucleotides that provides the starting point for DNA synthesis. In a PCR reaction, the experimenter decides the region of DNA that will be copied or amplified by the primer she or he chooses. PCR primers are short pieces of single-stranded DNA, usually around 20 nucleotides in length. Two primers are used in each PCR reaction, and they are designed so that they flank the target region. That is, they are given sequences that will make them bind to the opposite strands of the template DNA, just at the edges of the region to be copied. The primers bind to the template by complementary base pairing. When the primers are bound to the template, they can be extended by the polymerase, and the region that lies between them will get copied. Both primers, when bound, point inward, that is, in the 5' to 3' direction towards the region to be copied. Like other DNA polymerases, Taq polymerase can only synthesize DNA in the 5' to 3' direction. When the primers are extended, the region that lies between them will thus be copied.
The key ingredients of a PCR reaction are Taq polymerase, primers, template DNA, and nucleotides, which are DNA building blocks. The ingredients are assembled in a tube along with cofactors needed by the enzyme and are put through repeated cycles of heating and cooling that allow DNA to be synthesized. The basic steps are denaturation. You heat the reaction to about 96 degrees Celsius to separate or denature the DNA strands. This provides single-stranded template for the next step. Step 2 is annealing. You cool the reaction to about 55 through 65 degrees Celsius so the primers can bind to their complementary sequences on the single-stranded template DNA. Step 3, extension. Raise the reaction temperature to 72 degrees Celsius so Taq polymerase extends the primers, synthesizing new strands of DNA. This cycle repeats 25 to 35 times in a typical PCR reaction, which generally takes 2 to 4 hours depending on the length of the DNA region being copied. If the reaction is efficient, the target region can go from just one or a few copies to billions. That's because it's not just the original DNA that's used as a template each time. Instead, the new DNA that's made in one round can serve as a template in the next round of DNA synthesis. There are many copies of the primers and many molecules of Taq polymerase floating around in the reaction, so the number of DNA molecules can roughly double in each round of cycling.
Using PCR, a DNA sequence can be amplified millions or billions of times, producing enough DNA copies to be analyzed using other techniques. For instance, the DNA may be visualized by gel electrophoresis, sent for sequencing, or digested with restriction enzymes and cloned into a plasmid. PCR is used in many research labs, and it also has practical applications in forensics, genetic testing, and diagnostics. For instance, PCR is used to amplify genes associated with genetic disorders from the DNA of patients. PCR can also be used to test to test for a bacterium or DNA virus in the patient's body. If the pathogen is present, it may be possible to amplify regions of its DNA from a blood or a tissue sample.
The most commonly used type of markers in forensics, called short tandem repeats, or STRs, consist of many repeating copies of the same short nucleotide sequence, typically 2 to 5 nucleotides long. One allele of an STR might have 20 repeats, while another might have 18 or just 10. By examining multiple markers, each of which comes in many allele forms, forensic scientists can build a unique genetic fingerprint from a DNA sample. In a typical STR analysis using 13 markers, the odds of a false positive, or two people having the same DNA fingerprint, are less than 1 in 10 billion. Although we may think of DNA evidence being used to convict criminals, it has played a crucial role in exonerating falsely accused people. Forensic analysis is also used to establish paternity and to identify human remains from disaster scenes.
Polymerase chain reaction, or PCR, is a technique used to make many copies of a specific DNA region in vitro. PCR relies on a thermostable DNA polymerase, Taq polymerase, and requires DNA primers designed specifically for the DNA region of interest. In PCR, the reaction is repeatedly cycled through a series of temperature changes, which allows many copies of the target region to be produced. PCR has many research and practical applications. It is routinely used in DNA cloning, medical diagnostics, and forensic analysis of DNA.